Hello everyone, welcome to my channel Chem Insider. In the previous video of this particular class 11 chapter, we discussed about the method of purification of organic compounds if those organic compounds are present in solution states, liquid states, and they are mixed with through a non-volatile liquid or a volatile liquid. That is the method of distillation. Now we'll discuss the fourth method of purification, that is differential extraction. Yes, this particular differential extraction method is based on the physical property of extent of solubility. Now, whenever you consider for an organic compound which is soluble in water, but it is also soluble in organic solvent in a greater extent. Like for example, if an organic compound is soluble in water and also in an organic solvent, there will be clearly a difference in the solubility extent. Like for example, the organic compound may be soluble for about 5 grams in 100 ml of water, and it may be soluble in 50 grams to 100 ml of organic solvent. Then those type of organic solutions those type of organic compounds can be purified this by using this particular purification method. In this particular method, what we do is we take two specific solvents which are clearly immiscible to one another. Like for example, a water and an organic solvent are immiscible to one another. Usually the solvent which we use in organic compounds are ether or aniline. These two usually are not immiscible with one another. Water is not immiscible with ether, so therefore they will be forming a separate layer. Now what we do is we'll take the organic compound in a separating container like this. This is a separating container for extraction. So in this particular container we'll add the organic compound which is already soluble in certain different compound. Let us say it is soluble in water but for a lower extent. Now let us say an organic compound A is soluble in water while we are preparing it, and we have taken the particular compound added to this separating funnel, and later we are adding our organic solvent to it. Now since the organic solvent or the organic compound has a greater extent of its solubility in the organic solvent, it will start to dissolve in the organic solvent than that of the water. Therefore what we do is we'll add these three particular components and shake this separating funnel in a hard manner. So we'll shake this thoroughly in such a way that this mixes one with one another in a complete fashion to form almost a homogeneous mixture, even though it is a heterogeneous mixture. Once that is completely mixed thoroughly, what we do is we set it aside for a few minutes by opening the upper cork. So this upper cork helps us this particular compounds to form a layer from one above the other. The one which is denser will be forming a bottom layer, and the one which is lighter will be forming a higher upper layer. Okay. So now if you consider water is collected at the bottom, if you consider water is being collected at the bottom, we will separate water by removing the lower cork and such until on which we have a maximum amount of water being removed. Later we will further add some more amount of the organic solvent and stir it again, and we will remove the excess of unwanted solvent present in the separating funnel. Once you have completely separated the unwanted solvent, we will take the mixture of organic solute dissolved in a particular organic solvent and do a fractional distillation. Thus we can obtain a purified component of organic compound. Now the situation what we come across in this particular type of extraction purification process is most of the compounds are not completely soluble in organic solvents. So in those type of cases, what we do is we carry out multiple process of mixture and separation, which is called continuous extraction. Okay. In some cases, the method of if you repeat the cycle for 2 or 3 times, it will be more than enough. But in some cases, due to the very low extent of solubility, we have to carry out the particular repetition process for more than 10 to 15 cycles. Hence, those type of purification of extraction process are called continuous extraction process. Okay, so this is all about the differential extraction process. Next is the last one, chromatography techniques. So these chromatography techniques are quite very important in drugs chemistry. If you are preparing certain organic compound in a purified manner, or if you have to check the purity of an organic compound which you have prepared, then we use this particular method of purification, that is called the chromatography technique. So there are different types of chromatography technique depending upon the principle we use for this particular chromatography. So what happens in a chromatography process? The chromatography process involves three particular components. One is a stationary state, stationary phase, stationary absorbent. One we have a stationary absorbent, and second one we have a mobile phase, and third one is the solvent which you have to separate, solvent or solute or organic compound which you have to separate through the chromatography technique. So now what happens here is, we'll take a medium of mobile phase. So this particular mobile phase has a motion involved in it. So what we do is, when you take a sheet of paper or a column chromatography technique, we use this particular solute taken in this particular mobile phase of a component, and we dip that in the stationary adsorbent in such a way that it flows from one direction to the other direction in a slow process, and due to the presence of variable adsorbent constants for particular solvents or solutes, we observe different layers being accumulated in the containers. Thus, we can clearly separate those particular compounds using their color. So, why do we name this chromatography? Is in German, the name chroma means color. So, this is a technique which was developed by the Germans in order to purify organic compounds using their specific colors. So, there are two types of chromatography techniques. One is adsorption chromatography technique, and the other one is partition chromatography technique. One is adsorption chromatography technique, and the other one is partition chromatography technique. Now first we'll start discuss about the adsorption chromatography technique. So this adsorption chromatography technique is a process where the solute or solvent or an organic compound which you have to purify is adsorbed on a specific adsorbate. Okay. The first one is adsorption chromatography. So this adsorption chromatography are of two particular methods. One is column chromatography and the other one is thin layer chromatography. One is column chromatography and the other one is thin layer chromatography. So what do we do in a column chromatography technique? So if you take a column of container like this particular glass container will be having a woolen or cotton bud at the bottom to stop the flow of adsorbate. So here we have three particular components, as I said earlier itself. One is a stationary adsorbent, it will be filled at the bottom. We'll fill the stationary adsorbate at the bottom, and above it we'll be having an adsorbent. We'll be having a component which is to be separated or purified, adsorbed on the surface of the adsorbent. Okay, so this is the stationary adsorbent. Above this stationary adsorbent, we'll be having our compound which is to be purified, mixture of a compound which is to be purified, adsorbed on this particular surface of the adsorbent. So this is the mixed compound. Now, once this is been done, next what we'll do is, we'll take one more adsorbent, that is another adsorbent which is useful to get these mixed compound to adsorb in themselves. But the extent of adsorption of these mixed components is different for different components present in it. Let us say if it has component A, B, C in it, A will have a different adsorption ratio, B will have a different adsorption ratio, C will have a different adsorption ratio. Now, once you have filled this particular component with the mixed compound and the stationary adsorbent, next we'll add the mobile adsorbent, that is the adsorbent which will flow through this particular column in slow motion. So, once you add the mobile adsorbent, what we observe is A, B, C, all these three get adsorbed on the freshly added new adsorbent. This adsorbent flows through this particular stationary adsorbent in such a way that the mixed component ABC will be varying their adsorption process and will be collected in three different stages. If you have passed the solution in this particular column after a period of time, one of the component will be collected over here, one will be collected over here, and one will be present over here. So now this is the adsorbed solution. This is the stationary adsorbent, and in this stationary adsorbent, due to the flow of our mobile adsorbent, this particular levels of ABC will be formed, and this ABC will be separated later in order to purify this particular components present in the mixture of this compound. Okay, so this is the process of extraction or purification of organic compounds which are adsorbed, which have a different adsorption ratios. So this particular method which we are using for a column glass tube, hence it is called the column chromatography technique. Okay, so this is the first type of adsorption process. Next is thin layer chromatography. Thin layer chromatography is quite easier when compared to the column chromatography, but what we do is, in this particular condition, we use a thin thin paper strip which has a permeable membrane in it, and another particular thin layer of a membrane strip will be taking our compounds in a dot form. Okay, so if you consider this as our thin paper strip or the membrane strip, so this thin membrane strip will take the mixed compound which has to be purified on this strip as a certain dot. It is having certain mixture of compounds, let us say different colors of white, red, orange, green, red, etc. And then we will be drawing a line at the bottom of this strip called the baseline. This particular line is called the baseline, and this dot is called the compound dot. Okay. So here, this particular thin layer strip is the stationary phase. This is the stationary phase. That means it will be not moving. Next, what we'll do is, we'll dip this thin membrane strip in a glass container or a glass cap in such a way that the baseline touches the solution of the mobile phase, just like that. Like for example, if you consider this as the container, and we'll fill the mobile phase solution inside it in such a way that it is just touching the baseline. Okay, so it should be just touching the baseline of the mobile strip. So if you consider this as a container, we'll place the strip in such a way that the baseline should be just touching the solution. So this particular process takes a longer duration of time. That is because in the column chromatography technique, we observe the mobile phase moving from upwards to downward direction. But in this particular thin layer chromatography, what we do is, we take this particular container in a closed glass jar. We keep this in a closed glass jar which is filled with the eluent. An eluent is a compound which increases the adsorption of compounds, increases the adsorption of these particular compounds, which are taken as a dot on the thin strip onto the solvents. Okay. So now, after some period of time, the solvent which is taken in the glass container start to move in the upward direction. Okay. The solvent or the mobile phase of liquid which is taken in the glass plate moves upward in such a way that it also carries the liquid present here. For this particular example, you can just try taking water in a glass, a paper plate, and try to dip the paper sheet in it in a slight touching manner. What you observe is water will be flowing through the paper, right? Water will flow through the paper from downward to the upward direction. So similarly, this particular paper strip of thin layer will be gaining this particular solvent inside it, and that solvent gets absorbed through the thin layer paper. Once the solvent is moving in the upward direction, it will also carry these compounds present in the dot taken on the thin layer. So after some period of time, what we observe is the dot which was taken inside the glass container dipped in a glass plate has been splitted into multiple layers. So if this is the liquid which is filled through this, so let us say if liquid has been filled up to this much on the thin paper strip, you will have an orange color over here like this. You will have a red, green, blue, or a yellow color separated like such way. Okay, the compounds which are colored in nature will be split in a clear manner. So this is how a thin layer chromatography technique works. The problem over here is if you have colored compound, it can be clearly visible to your naked eye. But if you have certain colorless organic compounds, then we have to take those colorless organic compound strip under the influence of ultraviolet rays. When you read those thin membrane strips under the ultraviolet lights, you can clearly observe certain other components which are colorless in the visible region. Okay, or you can also use, instead of eluent, you can use an iodine spray in order to understand the colorless compound being adsorbed. What happens is when you spray iodine on this thin layer sheet, the compounds which are colorless in the visible region will be turning out brown once they are adsorbed due to the iodine present in the glass container which is placed on top of this particular constant, okay, the particular apparatus. Okay, so this is the method of thin layer chromatography technique. I hope you have understood this particular adsorption chromatography. In the next video, we'll discuss about partition chromatography technique. Okay, so this is all about for today. I hope you have understood these concepts. If you have, please do subscribe to my channel and share this video among your friends. Thank you for watching.